Krebs cycle is very important cycle. The Krebs cycle named in honor of the Hans Krebs, who worked out the intermediate step in this pathway, also known as the citric acid cycle or tricarboxylic acid cycle, is the second of the three pathways involved in the fuel catabolism and ATP production. It utilizes the molecular fragments formed during the carbohydrate, protein and fat breakdown, and it produces carbon dioxide, hydrogen atoms bound to coenzyme and small amounts of ATP. The enzyme for this pathway are located in the mitochondria. Most of these enzymes are soluble enzymes in the inner mitochondrial compounds. The primary molecule entering at the beginning of the Krebs cycle is acetyl coenzyme A. Coenzyme A, a precursor of acetyl coenzyme A, is derived from the B vitamin pantothenic acid, and its primary functions is to transfer the two carbon acetyl group from one molecule to another. This acetyl group comes either from pyruvate, which as we have just seen in the end product of glycolysis, or from the breakdown of the fatty acids and some amino acids, as we shall see in the later section. Pyruvate, upon entering mitochondria from the cytosol, undergoes the following reaction to produce acetyl coenzyme A. Pyruvate plus coenzyme A plus NAD ion is equal to acetyl CoA plus CO2 plus NADH plus H ion. Pyruvate, this is pyruvate, plus coenzyme A plus NAD ion is equal to acetyl coenzyme A plus CO2 plus carbon dioxide plus NaDH plus hydrogen ion. Pyruvate plus coenzyme A plus NAD ion is equal to react acetyl coenzyme A plus carbon dioxide plus NaDH plus hydrogen ion. Here in this equation, hydrogen ion released, pyruvate plus coenzyme A plus NAD ion reacts and make acetyl coenzyme A and produce carbon dioxide, NaDH plus hydrogen ion. Note that this reaction produced the first molecule of carbon dioxide we have seen formed thus far in the pathway of fuel catabolism, and that hydrogen atoms have been transferred to NAD. hydrogen atoms transfer to NAD. These are the hydrogen ion can transfer to NAD. This is the hydrogen ion with NAD. We will make use of this later fact later. The Krebs cycle begins with the transfer of acetyl group of acetyl coenzyme A to 4 carbon molecules. Oxaloacetate to form the 6 carbon molecules. Citrate. At the third step in the cycle, 1 molecule of carbon dioxide is produced, and again the fourth step, thus 2 carbon atoms entered the cycle in the form of the acetyl group attached to coenzyme A, and now 2 carbons, although not the same one, have left in the form of carbon dioxide. Note also that the oxygen that appears in the carbon dioxide is not derived from molecular oxygen but from a carboxyl group of Krebs cycle intermediates. In the remainder of the cycle, the 4 carbon molecules formed in reaction 4 is modified through a series of reactions to produce the 4 carbon molecules oxaloacetate, which become available to accept the another acetyl group and repeat the cycle. Now we come to a very crucial fact. In addition to producing carbon dioxide intermediate in the Krebs cycle, donate hydrogen atom to the coenzyme NAD ion and FAD. 2 hydrogen atoms are transferred to NAD ion in each of step 3, 4 and 8, and to FAD in reaction 6. These hydrogen will be transferred from coenzyme to oxygen in the next stage of fuel metabolism. Oxidative phosphorylation. Since oxidative phosphorylation is necessary for regeneration of the hydrogen free form of these coenzymes, the Krebs cycle can operate only under aerobic conditions. There is no pathway in the mitochondria that can remove the hydrogen from the coenzyme under the anaerobic conditions. So far we have said nothing of how the Krebs cycle contributes to the formation of ATP. In fact, the Krebs cycle directly produces only one high energy nucleotide triphosphate. This occurs during reaction 5, in which an organic phosphate is transformed to guanosine diphosphate GDP to form guanosine triphosphate GTP. The hydrolysis of GTP, like that of ATP, can provide energy for some energy requiring reactions. In addition, the energy in GTP energy requires reaction. In addition, the energy in GTP can be transferred to ATP by reaction. GTP guanosine diphosphate plus ADP adenosine diphosphate is inversely proportional to GDP plus ATP. GDP, guanidine diphosphate plus ATP is a reversible action, GDP plus ATP. This reaction is reversible and energy is in ATP can be used in the form of GDP. We can say GDP plus ADP, guanidine triphosphate plus adenosine diphosphate ADP is a reversible reaction in GDP, guanosine diphosphate, guanosine diphosphate plus adenosine triphosphate. Adenosine triphosphate plus GDP can make a reaction and release energy with GDP plus ADP. This reaction is reversible and the energy in ATP can be used to form GTP from GDP. One additional GTP is required for this molecule's role in protein synthesis and signal transduction. Reiterate the formation of GTP is the only mechanism by which ATP is directly formed within the Krebs cycle. Why then is the Krebs cycle so important? Because the hydrogen atoms transferred in the cycle to the concerned enzyme are used in the most pathway oxidative phosphorylation to form large amount of ATP. The net result of the catabolism of one acetyl group from acetyl coenzyme A by way of the Krebs cycle can be written as acetyl coenzyme A plus 3 NADIN plus FAD plus GDP plus PI plus 2 water molecules is equal to 2 carbon dioxide plus coenzyme A plus NADH plus 3G. We can understand with this equation acetyl coenzyme A plus NADN plus FAD plus FAD plus GDP plus GDP plus PI plus 2 water molecules are react and make 2 carbon dioxide ions, 2 atom of carbon dioxides plus coenzyme A plus 3 NADH plus 3 NADH plus 3 hydrogen ion plus FADH2 plus FADH 2 plus GDP plus GTP. This is the chemical reaction. We can understand this reaction: acetyl coenzyme A plus 3 NAD ion plus FAD, acetyl coenzyme A plus NAD ion plus FAD plus GDP plus Pi plus 2 hydrogen atom, 2 H2O is equal to 2 carbon dioxide plus coenzyme A plus NADH plus 3 hydrogen ion plus FADH2 plus GTD. This is not DGD, this is the DGP. One more important should be noted: although the major function of the Krebs cycle is the provision of the hydrogen atoms to the oxidation phosphorylation pathway, some of the organic molecules, especially several types of amino acids required by cell, oxaloacetate is one of the intermediate used in this manner. When a molecule of oxaloacetate is removed from the Krebs cycle in the process of forming amino acid, it is not available to combine with the acetate fragment of acetyl coenzyme A at the beginning of the cycle. Thus, there must be a way of replacing the oxaloacetate and other Krebs cycle intermediate that are consumed to synthetic pathway. Carbohydrate provide one source of oxaloacetate replacement by following reaction, which convert pyruvate into oxaloacetate. Contrast this reaction to equation pyruvate plus carbon dioxide plus ATP, pyruvate plus carbon dioxide plus ATP oxaloacetate plus ADP oxaloacetate plus ADP plus Pi. Oxaloacetate plus ADP plus Pi. Certain amino acids derived as well C also can be used to form oxaloacetate and other Krebs cycle intermediate. Oxidative phosphorylation. Oxidative phosphorylation provides the third and quantitatively most important mechanism by which energy derived from fluid molecules can be transferred to ATP. The basic principle behind the pathway is simple. The energy transferred to ATP is derived from the energy released when hydrogen combines with the molecular oxygen to form water. The source of hydrogen is the NADH plus hydrogen ion and FADH2 plus coenzyme molecules. The net reaction involving NADH plus hydrogen ion is half oxygen plus 88 H2O plus NAD53. 1 mole of ATP from ADP and PI, there is enough energy released in the above reaction to form more than 1 molecule of ATP. The protein that mediate oxidative phosphorylation are located in the mitochondria, but unlike the enzymes of the Krebs cycle, which are soluble enzymes in the matrix compartment, the proteins that mediate oxidative phosphorylation are embedded in the inner mitochondrial membrane. These proteins can be divided into two groups. Number one, those that mediate the series of reactions by which hydrogen is transferred to molecule oxygen, and number two, those that couple the energy released by these reactions to the synthesis of ATP. Most of the first group of proteins contain iron and copper cofactor, forming proteins known as cytochromes. Because in pure form they are brightly colored, they resemble in their structure the red iron containing hemoglobin molecule which binds and the oxygen in the blood. The cytochrome forms the compound of the electrons transport chain, in which two electrons from the hydrogen atoms are initially transferred either from NADH plus hydrogen ion plus FADH2 to one of the element in the electron. The transport chain, these electrons are then successively transferred to other elements in the chain, often to or from an iron or copper ion, until the electrons are finally transferred to molecule oxygen, which then combines with hydrogen ion proteins to form water. These hydrogen ion, like the electrons, come from the hydrogen bearing coenzyme having been released from them early in the transport chain when the electrons from the hydrogen atoms are transferred to the cytochromes. In addition to transfer the coenzyme hydrogens to water, this process regenerates the hydrogen free from the coenzymes, which then becomes available to accept two more hydrogen from intermediary reactions. Thus, the electrons transport chain provide the aerobic mechanism from regenerating the hydrogen free form of this coenzyme, whereas the described earlier the aerobic mechanism is coupled in the formation of lactate at each step. Along the electron transport chain, small amount of energy are released, which in total amount to the full 53 kilocalorie per mole released from a direct reaction between hydrogen and oxygen. Because this energy is released in small steps, it can be linked to the synthesis of several molecules of ATP, each of which requires only 7 kilocalorie per mole. Overall, 40% of energy released from the chain of reactions is transferred to ATP, the remaining 60% appearing as heat. At 3 points along the electron transport chain, ATP is formed. The mechanism by which this occurs is known as the chemiosmotic hypothesis, the electrons are transformed along the cytochrome chain. The energy released is used to move hydrogen ions from the matrix to cytocyclic side of the inner mitochondrial membrane, thus producing a source of potential energy in the form of point. Along with the chain complex of protein forms a channel through which the highly concentrated hydrogen ions on the cytoplasmic side of membranes can flow back to the matrix side, and the process transfer their potential energy to formation of ATP from ADP and PI. Metabolism, the sum total of chemical processes that occurs in living organisms, resulting in growth, production of energy, elimination of waste materials, etc. And anabolism is that build-up of complex molecules. Next is catabolism. Catabolism means breakdown of complex molecules. Fate of organic building block in ATP metabolism. Organic building block molecules, monosaccharides, amino acids, acetate, nucleotide base, ATP and ADP plus PI make energy. This is the catabolism, and when produce energy called anabolism, and when energy breakdown from ATP to ADP plus PI is called catabolism, and this produces carbon dioxide and H2O. And in condition of anabolic process, polymers and other energy rich molecules are produced. Cellular respiration, C6H12O6 plus 6 oxygen plus 36 ADP plus 36 PI is equal to 6 CO2 plus 6 H2O plus 36 ATP. This in the cell chemical reactions, C6H12O6 plus 6 CO2 plus 36 ADP plus 36 PI reacts with each other and make carbon dioxide plus water plus 36 ATP. This release energy in the form of ATP. Basic steps involved in the metabolism. Number 1, glycolysis. Glycolysis means the breakdown of glucose. Lysis means breakdown. And next step is acetylcholine A formation. And third step is Krebs cycle. And fourth step is electron transport system. Whichever electrons in a ion condition and transported, this is known as electron transport system. Overview of glycolysis. When glucose break down in ATP and NADH plus H2 made pyruvic acid and 2-acetyl coenzyme A, formation of acetyl coenzyme A, and break down into CO2, NADH plus 2 hydrogen. In next step, Krebs cycle, in a cycle NADH plus hydrogen, FADH2 and other electrons in a gut electrons transport chain, when different electrons transported oxygen and hydrogen and produce energy in 32 or 34 ATP. Other metabolic pathway: number 1 fat, number 2 glycogen, number 3 protein. We can take first fat. Fat break down into two stages: fatty acid and glycerol. Fatty acid further reach in acetyl group in the cells and involve in Krebs cycle and produce 2 ATP with the help of carbon breakdown, pyruvate, and also glycerol. Electron transport system, many ATP releases. We can take glycogen also break down in the glucose, and pyruvate, which is derived from glycerol and also from glucose, and break down in carbon breakdown and make acetyl group and reached in Krebs cycle and produce energy. And transport, electron transport system may make and many energies molecules. We can take third proteins. Proteins make amino acid and carbon breakdown, NH3 ammonia, urea waste, produce urea as a waste product. But carbon breakdown involve in acetyl group and help in Krebs cycle and also produce ATP. Chemical energy: high energy electrons, glycolysis, glucose when breakdown known as glycolysis, it breaks in pyruvate and reach to Krebs cycle, and in the chemical changes and chemical energies, electron transport chain and oxidative phosphorylation, in presence of within mitochondria. Why the oxidative phosphorylation? This release ATP, and also from Krebs cycle ATP produced, and cytosol mitochondrial crystal via this substrate level phosphorylation ATP also produced in this case. Glycolysis: first of all, glucose breakdown in 2 ATP and 2 ADP, and in next stage 2 PGAL and further breakdown in 2 NAD and 2 NADH and 4 ADP and 4 ATP. And next, in the next step of with chemical reactions, 2 pyruvate formation and carbon releases. 2 ATP, 2 NADH, 2 pyruvate molecules releases and produce energy. Balance sheet for glycolysis: input glucose 2 ADP plus Pi plus NAD ion, output pyruvate ATP and NAD. 2 pyruvate, 2 ATP and 2 NAD. Transaction reaction: cytosol transport proteins, pyruvate, and in mitochondrion NADH and hydrogen ion, carbon dioxide release, coenzyme A also produce, and further acetyl coenzyme A formation. Transition reaction: Krebs cycle citric acid. Transition reactions: citric acid cycle, as early I described in the equations and the formulations. Citric acid cycle: 3 NAD, 3 NAD cycle, FAD plus FADH2, 2 carbon dioxide release, ADP, produce ATP. In citric acid cycle, coenzyme A, carbon releases acetyl coenzyme A, carbon dioxide releases NADH plus NAD ion and make pyruvate, ultimately release energy in form of ATP. Krebs cycle, citric acid cycle. Next is the balance sheet for the transition of reaction of Krebs cycle. Input is 2 pyruvate, 2 ADP plus 2 PI, 8 NAD ion plus 2 FAD, and the output of this chemical reaction is 6 carbon dioxide, 2 ATP energy, 8 NADH and 2 FADH2. Krebs cycle. We can describe multiple times in the Krebs cycle is a complex and complicated cycle, handles other substrate, intermediate molecules used, proteins and lipids, replenishment of intermediate necessary acetyl coenzyme A, oxalic acid formation, and ultimately produce energy. Krebs cycle take 2 complete cycles, 8 steps, each with an enzyme. Oxaloacetic acid acetyl coenzyme A, and acetyl coenzyme in 3 steps, and third step we can say this is the third step, oxaloacetic acid, NADH, FADH2 and the citric acid, carbon dioxide release, NADH and ATP produce CO2 and NADH. This cycle is a circular cycle producing acetyl coenzyme A and citric acid and releases carbon dioxide and produce ATP, oxaloacetic acid. Oxidative phosphorylation described earlier, each glucose molecules, carbon dioxide 6 molecules, NADH 10 molecules, FADH 2, ATP 4 molecules. Electron transport system. CO2 6, NADH 10, FADH 2, ATP 4 used to make ATP. NADH and FADH 2 produce energy with the help or with the main function of ATP. Electron transport system. Proton discharge electron receptors generate ATP. This is the fourth step of the Krebs cycle. Electron transport system. NADH, NAD, hydrogen ion, and in the lumen hydrogen ion 2H, half oxygen and water release in the mitochondria and matrix. In intermembranous spaces, hydrogen ion releases. And next condition, mitochondrial matrix, intermembranous spaces, hydrogen ion reaches, many hydrogen ion reaches in the intermediate membrane spaces, and in mitochondrial matrix, hydrogen ion, ADP plus PI, ATP and hydrogen resided, and this ATP produce energy. Electron transport chain and oxidative phosphorylation. Electrons are delivered to O, forming O attracts hydrogen ion to form H2O. Electrons are delivered to O, forming O negative. This is the oxygen ion. Oxygen ion with the help of hydrogen ion to form H2O. Electronic energy gradient transfer to energy from NADH plus hydrogen ion and FADH2. Oxygen releases large amount of energy This energy is released in a stepwise manner through the electron transport chain. ATP synthesis. Two major parts connected by a rotor in the inner mitochondrial membrane, knob in the matrix, works like an ion pump in reverse. Net ATP yield: 34 to 36 molecules ATP for every glucose molecules produced, about 40% of efficiency. ATP yield from the basically glucose breakdown, other we can say the glucose molecules produce ATP in 34 to 36 molecule ATP for every glucose molecules, or otherwise we can say one glucose molecule produce 34 to 36 ATP molecules and produce energy. Overall ATP production: electron transport system 34, citric acid cycle 2, glycolysis 2, subtotal 38, NADH transport into mitochondrion -2, total is 36. Fermentation. Fermentation is the anaerobic respiration. Lactic acid fermentation. Lactic acid fermentation, in case of lactic acid fermentation, glucose breakdown, we can say this is the glycolysis and make NADH and NADH ion, NAD ion and NADH ion, and make 2 pyruvate, and further it is NADH and NAD ion, lactic acid fermentation, 2 lactic acid produced in this condition. Glucose breakdown, glycolysis, NAD ion and NADH ion. NAD ion plus NADH ion produce lactic acid fermentation and produce 2 lactic acid with the help of 2 pyruvate. Glucose. Anaerobic respiration, aerobic respiration. What is glucose? Glucose is the basic. Glucose in anaerobic condition makes pyruvate in both condition and NO2, ethanol or lactate. Aerobic respiration with the oxygen acetyl coenzyme with the help of Krebs cycle in mitochondria.